Hello everyone, today I will be discussing about transgenic animals. So, what is transgenic animal? A transgenic animal is an animal that has been genetically engineered to contain one or more extra gene in its cell. It now has a new inheritable property of some type. Genetically modified organisms are organisms with artificially altered DNA, that is created by adding a foreign gene altering the base sequence of an existing gene. Adding a foreign gene, if we add the foreign gene, production of insulin and vaccine for altering an existing gene is an organism are altered to make it produced at higher levels, that is growth hormone. For construction of transgene, a transgene must contain the gene of interest along with extra DNA sequences, that is promoter sequence and sequence of termination, which control the function of the foreign gene in the transgenic animal. Three parts are required to construct a transgene: a tissue specific promoter sequence that determines the tissue specific function. For example, if the gene needs to be expressed in milk of mammal, the promoter specific for mammary tissues is used in the transgene. Second one is the sequence of the gene coding for amino acid of the desired protein, and the third one is the sequence responsible for the termination of the expressed protein. For purification of transgenic DNA constructs, a sucrose gradient or a gel purification method can be adopted to obtain purified and clean DNA that is dissolved in a microinjection buffer. So, methods of creation of transgenic animals: first is retrovirus mediated transgenesis, second one is pronuclear microinjection, third one is embryonic stem cell method, and fourth one is the nuclear transfer. So, in retrovirus mediated transgenesis, retroviruses are commonly used as vector to transfer genetic material into the cell, taking advantage of their ability to infect host cell in this way. Offspring derived from this method are chimeric, that is not all cells carry the retrovirus. Transmission of the transgene is possible only if the retrovirus integrates into some of the germ cells. So, in this pictorial representation, you can see the retrovirus mediated transgenesis. Next technique is the microinjection. Eggs are harvested from super ovulated animals and fertilized in vitro. 200 to 300 copies of the foreign genes are injected into the recently fertilized egg, implanted into surrogate mothers. A small proportion of the animals born are transgenic. So, this is the whole process you can see in this pictorial representation step by step. Then embryonic stem cell transfer, target gene sequences to specific sites in the genome, then alternation made to stem cell in culture, then mutated stem cell injected into the blastocyst, and then blastocyst implanted into the foster mother. Embryonic stem cell, you can see here the first cell stage, then 2 cell stage, then 4 cell stage and 8 cell stage here, and then blastocyst, then fetus and adults. Then nuclear transfer, next technique, removing the nucleus of an unfertilized egg and replacing it with the nuclear transfer. Removing the nucleus of an unfertilized egg and replacing it with the nucleus of a donor cell An electric current is used to fuse the nucleus in the cell and trigger the egg to begin developing. The reconstructed eggs are then implanted into a surrogate mother. Somatic cells can be cultured, subjected to genetic manipulation in vitro and then produce viable animals by means of nuclear transfer. In this pictorial representation, you can see the genetic engineered or transgenic animals. Improving agriculture products with transgenics, faster growth rates or learner growth pattern, improve the product or more product, increase nutritional content lactoferrin, turning the animals into efficient grazers, transfer antimicrobial genes to farm animals. Here you can see the whole process in this pictorial representation. Transgenic animals as a bioreactor. Biosteel, otherwise known as spider silk, cloned into goat milk, that is commonly known as silk milk goats. Goats reproduce faster than cows and are cheaper than cows. Hence, also make good bioreactor in that they are cheap and a lot of eggs are produced at one time. Producing human antibodies in animals, production of monoclonal antibodies used to treat cancer, heart diseases and transplant rejection. Humanized monoclonal antibodies were developed to prevent the human anti-mouse antibody response. Here you can see the whole process. Then knockout, a special case of transgenesis. A specific gene is disrupted or removed such that it is not expressed. DNA is modified, it is added to embryonic stem cells and where it undergoes homologous recombination. The modified E cells are then introduced into normal embryos and then embryo is implanted in an incubator mother. The offspring is a chimera. Some of the remarkable products developed through transgenesis in animals that include production of human proteins in animal milk, production of biosteel, a high strength silk product from goat's milk, growth of tissues on 3D printer by a genetic manipulation of stem cells can be used as skin substitute for wound healing, production of therapeutic proteins such as monoclonal antibodies from the milk of transgenic cows, goat and mice, which is used to administer drug in various diseases. Example of transgenic animals are transgenic fish, that is superfish that increase growth and size, growth hormones gene insert into the fertilized egg, and transgenic salmon grow about 10 to 11 times faster than normal fish. You can see the length of the transgenic fish here, that is 10 to 11 times larger than glowfish. Next example is the glowfish, that is Danio rario. Genetically modified freshwater zebrafish or Danio rario that is produced by integrating a fluorescent protein gene from jellyfish into embryo of fish. You can see here the fluorescent color, different color, red fluorescent color, green fluorescent color and yellow fluorescent fish here, that is transgenic fish. Then transgenic mouse, the Alzheimer mouse. In the brain of Alzheimer's patients, dead nerve cells are entangled in a peptide called amyloid. Mouse made by introducing amyloid precursor gene fertilized egg of mice. Next example is the onco mouse, mouse model to study cancer made by inserting activated oncogenes. Next example is the smart mouse, biological model engineered to overexpress NR2B receptor in the synaptic pathway. This make the mice learn faster like juveniles throughout their lives. You can see in this picture. Then transgenic pig and viropig. Pig have the trouble of fully digesting a compound known as phytate found in the many cereal grain used to feed them. Transgenic pig are created by the introducing phytase gene of E. coli. Then phytase enzyme is thus produced in the salivary gland of pig. It is degrade in digestible phytase with the release of phosphate that readily digested by pigs. You can see in this picture how we are feeding it and we are getting that enzyme. Pig for organ transplant, it is used for organ transplant. Pig with human genes in order to decrease the chances of organ rejection by human body. This is transgenic pig. Then transgenic livestock, bioreaction whose cells have been engineered to the synthesis marketable protein more economical than proceeding desired protein in the cell culture. Transgenic cattle, transgenic cows are made to produce protein lactoferrin and then interferon in their milk. Prior free cows resistant to mad cow disease. Transgenic sheep for good quality of wool production. Dolly was the first female domestic sheep and the first mammal cloned from somatic cell uses the process of the nuclear transfer from July 1996. She was the clone by the Sir and Sir Ian Wilmot, Keith Campbell and the colleagues and the Roslin Institute, the University of Edinburgh in the Scotland. Then transgenic goat, goat that could be expressed the tissue plasminogen activator and antithrombin 3 spider silk in milk. Then transgenic rabbit, Alba the EGFT enhanced green fluorescent protein bunny created in 2000 as a transgenic animal. Transgenic bunny, you can see here the green fluorescent transgenic bunny that is created in 2000. Then transgenic monkey, Andy was the first transgenic monkey born in 2000. Andy stand for inserted DNA spelled backward. An engineered virus was used to the inserted in the harmless gene for the green fluorescent protein into Andy's genome. Andy proved that transgenic primate can be created and can be expressed foreign gene delivered into their genome. Transgenic monkey Andy that is created in 2000. Then advantages. Advantages of the transgenic animals. At this increase growth rate, improve disease resistance, improve food conversion rate, it increases muscle mass also, it improves nutritional quality also and improve cell quality also. So, what are the disadvantages of the transgenic animals? Inserted gene has multiple function, breathing problems sometimes lead to mutagenesis and the function, solemn survival rate of the transgenic animals. So, these are the few advantages also. Then application of the transgenic animals. Transgenic animals are divided into the 5 classes based on their purpose. Disease model, transformers, genoplanters, food sources and biological models. So, these are the 5 purposes of application of the transgenic animals. We will take one by one. First, disease model. In disease model, animals that have been genetically altered to the express some aspect to the human disease, etiology of the complex disease, and to be developed the potential therapies without the use of the human subject. For example, AIDS mouse, Alzheimer mouse, onco mouse and other models, HTN, CAD, DM, etc. Transgenic models for HTN. Polygenic diseases, RAAS plays on important role. First model is the transgenic mouse, expressing rat renin and ongoing, that is gene is REN2 gene. In rat model, microinjection of mouse REN2 gene into oocyte of that rat, heterozygous transgenic rats, filament HTN rat as sensitive to ARBS or ACEIS, that is linino-pre. Then transgenic models of diabetes. For studying the genes and their role in peripheral insulin action, the model of insulin secretion is glucokinase and hepatic glucose production in the diabetes. So, in transgenic mouse model, IDDM by retrovirus factor method, if we see the mouse model for Alzheimer disease is two mutant genes of APP717 and APP670 or 671 were used for creation of transgenic mouse model for Alzheimer disease. The advantages of this model is the transgenes were introduced in the mice. The transgenic mice containing about 40 copies of the PDAPP mini gene or aging display amyloid plaques, neuronal cell death and memory defects. In Huntington disease an incurable fatal neurological genetic disorder. It remains confined to specific regions of the brain. About 1 in 10,000 people worldwide are affected by this disease. It due to the change in Huntington disease gene coding the Huntington protein, adding to CAG trinucleotides unit to exon 1 of the HD gene is responsible for the disease. During translation, CAG code for glutamine, consequently multiple CAG codons incorporate a series of glutamine residues, that is polyglutamine, in the Huntington protein or HD protein. The symptoms of Huntington disease occur when the number of CAG codons in glutamine region is 38 or more. Now, transgenic mouse model for Huntington disease. A mouse model for Huntington disease was created using theta conditional regulation system. HD genes that contain exon 1 with 94 CAG repeats was used as the transgene. The TTA gene was placed under the control of a promoter that is active in the cells of the forebrain. Expression of HD transgene was switched off in the embryos during pregnancy by adding doxycycline to the drinking water. After birth, doxycycline was not supplied to the transgenic mice. This allowed continuous expression of the mutant HD genes. A neurological conditions that was similar to the Huntington disease in humans was developed in these transgenic mice. The features of the disease disappeared when the expression of the mutant HD gene was prevented by the addition of doxycycline. This indicates that a continuous expression of the mutant HD gene is required for establishment of the disease. The brain cells can recover when this synthesis of HD protein occurs. You can see here in this pictorial representation the conditional expression of transgenic of. Next one is the transformer. Transgenic animals, which are the genetically engineered, the procedure of human pharmaceutical protein, recombinant protein in the saliva, milk, urine or blood. In 1920, insulin extracted from the pig pancreas. Early 1980s, human insulin prepared in recombinant bacteria. The synthesis of complex protein required for post translational modification to remain stable. Transgenic animal producing RPS, for example, in milk, human antithrombin 3, in chicken egg white, for example, MABS, human IFNS, HS, etc. Methods used are micro injection and nuclear transfer. For example, insulin for diabetes mellitus and vaccine. Next one is the xenotransplanters. Transgenic animals that are the genetically modified to have that can be transplanted into the human do not express key foreign antigens that are promising alternative to the human donors. Pig is the only animal used. Its physiology closely match from the human being. They are much less expensive than the monkey and other primates. Pig have alpha 1,3 galactose on the surface of their cell produced by the enzyme alpha 1,2 galactoside transferase. Next one is the food sources, creation of animal that grow larger without much food, more efficient, cost effective and food source, ethically and safety concern, not yet produced commercially as food source. Next one is the transgenic biological model, create to increase our knowledge about the function of protein by over expressing the gene encoding that protein or knocking it out in biological and genetic studies. For example, Andy, smart mouse and youth mouse. So, you have seen the transgenic animals, its application is advantage in disadvantages, but their lifespan is very short and the pig's physiology is very similar to the human being. So, we can introduce pig for organ transplant and any other diseases for genetic diseases we can use mouse and as I have discussed the fluorescent fishes. So, different transgenic animals we can produce genetically engineered, for example fish, mouse we can use for genetic diseases and pigs also for other diseases. So, genetically engineered animals are very useful also, having some disadvantages also. Thank you.